And now we move on to the lab diagnosis. In lab diagnosis, we shall discuss gram staining and culture. Specimen is obtained as indicated by the type of infection, like take specimen from skin infection, take pus from soft tissue infection, take urine in UTI, and take blood in sepsis, and take sputum in pneumonia, and so on and on. Gram stained specimen shows gram negative rod, that is motile and encapsulated. You can see these morphological features in this diagram. These morphological characters does not differentiate it from other enteric and from other gram negative rods. If you put the specimen under the electron microscope, you can see that it has polar flagella, and this polar flagella makes it mobile. It has pili. We can summarize the morphological features in this way, that the Pseudomonas aeruginosa is a motile gram negative encapsulated rod with polar flagella. It is gram negative motile encapsulated rod. It is gram negative rod, that is motile encapsulated, and it has polar flagella. Culture. Culture is the most specific test for the diagnosis of Pseudomonas aeruginosa infection. Pseudomonas aeruginosa is an obligate aerobe, and it grows on many types of culture media. Usually smear is plated on blood or other differential media, which are commonly used to grow enteric gram-negative rods. Pseudomonas aeruginosa grow readily on these media, but it grows more slowly than other ones, than other enterics. And one very important fact, please pay attention, Pseudomonas aeruginosa does not ferment lactose. So Pseudomonas aeruginosa grows as non-lactose fermenting colonies on McConkey or EMB agar. You can see in this McConkey agar plate with colonies which are colorless, and these colorless colonies indicate that this bacteria can't ferment lactose. This feature differentiates it from other lactose fermenting gram negative enteric rods. This is a really very important culture feature. Another important feature, Pseudomonas aeruginosa is oxidase positive. What does it mean that it is oxidase positive? And why it is oxidase positive? And what's the use of this test? Oxidase positive means that when reagent for the oxidase test is added to the colony, the colony which turns purple is oxidase positive. Like you can see in this picture, the left specimen is oxidase positive, as it has turned purple when the oxidase test reagent was added to it. The bacteria which are oxidase positive, they oxidize glucose. They do not ferment glucose. They oxidize glucose because they have a cytochrome oxidase C. And the bacteria which do not oxidize and do the fermentation of the glucose do not have cytochrome oxidase C. So this dark oxidized product is a result of cytochrome oxidase C. This test differentiates Pseudomonas from other oxidase negatives, gram negative rods. So Pseudomonas is a non-lactose fermenting oxidase positive gram negative rod. Please appreciate Pseudomonas is non-lactose fermenting oxidase positive gram negative rod. One more important fact that's unique with Pseudomonas. Pseudomonas aeruginosa produces diffusible bluish-green pigment, which is a combination of a pyoverdin and pyocyanin. Pyoverdin, yellow pigment; pyocyanin, blue pigment. This diffusible bluish-green color uh, gives a characteristic bluish-green color to the cluny, and it diffuses into the media. Okay, you can see this pigmentation in this agar plate. This pigmentation can be seen in agar around the Pseudomonas aeruginosa growth. Pseudomonas aeruginosa produces typical metallic sheen of growth on a blood agar plate, on a TSI agar plate. You can see this typical metallic sheen in this diagram, in this picture. Okay, another important feature of Pseudomonas aeruginosa growth. See this picture which shows the mucoid appearance of the Pseudomonas growth. It is due to the overproduction of alginate. Alginate is an exopolysaccharide. This exopolysaccharides provides the matrix for the Pseudomonas to live in this biofilm. And while living and proliferating in this biofilm, Pseudomonas causes the chronic lung infection in the patient in a patient with cystic fibrosis. So we can summarize the culture feature in this way, that the Pseudomonas is non-lactose fermenting, it is oxidase positive, it produces bluish green pigment, it has metallic sheen, it has mucoid appearance, and it has fruity aroma. And these features are sufficient to make the diagnosis of Pseudomonas aeruginosa, while the confirmation of the diagnosis is made with the biochemical tests. Again, Pseudomonas is non-lactose fermenting, it is oxidase positive, it has bluish green pigment, it has metallic sheen, it has mucoid appearance, it has fruity aroma, and these features are sufficient to make a presumptive diagnosis of the Pseudomonas. And the confirmation of the diagnosis is made with biochemical tests.